Hello dear learners, I welcome you all to the biochemistry tutorial session under the aegis of OneMedicine. This is the first tutorial on biochemistry and myself Dr. Sanghamitra Chakraborty is the guide by your side. To start with the first competency of biochemistry curriculum, that is, we will have a discussion on cell organelle and subcellular fractionation. Cell and the components of the cell. Majority of you must be thinking, cell, we know it from our school days. Yes, you know it from your school days, but we will have a small recapitulation of the organelle and subcellular components with their function as NMC have prescribed it is in our curriculum. So to start with this table, the first organelle, that is nucleus, and it is the site of chromosomal organization, replication and transcription. Next we have the mitochondria. This mitochondria, it is the site of various metabolic pathway like the citric acid cycle. It is the site of citric acid cycle, oxidative phosphorylation. The ribosome, the kitchen of the cell, it is concerned with protein synthesis or it is concerned with translation. Next we have the endoplasmic reticulum, that is also associated with protein synthesis or metabolism of the xenobiotics. Xenobiotics are any substance that are foreign to us, that may be toxins or any drug or any metabolite which is toxin produced as a byproduct of some metabolic pathway. Lysosome, they are the site of degradation reaction, that may be the degradation of proteins, enzymes, so on and so forth. Plasma membrane, it acts as a gateway that helps in docketing of substances from the cell to exterior and from exterior to interior of the cell. They also play a pivotal role in cell interaction, adhesion or signaling. Next we have the Golgi complex. They are mainly associated with the sorting of protein and glycosylation reaction. This Golgi complex, they functioned in glycosylation of protein, that means they make the protein biologically active. Whenever the protein is synthesized in the ribosome, some of the proteins in the naive form are not functionally that much active. To make them active or to send them to the targeted organ, this Golgi complex plays a key role. Next we have the peroxisome. The peroxisome are concerned with the branch chain fatty acid oxidation, degradation of hydrogen peroxide and free radicals. We have the cytoskeleton that are mainly associated with the microfilaments, tubules and intermediate filaments, and we have the cytosol that is associated with metabolism like glycolytic pathway and fatty acid synthesis. So this is a small and brief idea of cell with the organelle and subcellular components and their individual function. Next we have the key factor, that is subcellular fractionation. As the name suggests, it consists of two words, subcellular and fractionation. That means fractionating the cell into its component entities. So to start with the definition by book, the process of isolation of cell organelle in pure form and free of significant contamination by other organelle is called as subcellular fractionation. Albert Claude in 1930 pioneered the technique of subcellular fractionation and he identified the isolation of different organelle using centrifugation at very high speeds. Next, difference between organelle and subcellular components. Organelle are membrane bound and they are isolated by centrifugation at very high speed. Ribosome, cytoskeletal or filaments and cytosol, they are not membrane bound and they are not organelle. They are subcellular components but they are not organelle. Next, what are the key steps we are going to do for subcellular fractionation? Number one, we are going to go for extraction or homogenization. Next, we are going to go for centrifugation at very high speed, and lastly, we have to isolate and identify that okay, which organelle we have isolated from this particular cell. So first step is we have to break the cell, that is disrupt the cell. So the disruption of the cell can be done by two methods. Basically, one is the mechanical, another one is the non-mechanical. Mechanical can be done by shear stress on the cell. So that may be under two types, solid shear and liquid shear. Solid shear with the bead technology or bead meal technology, liquid shear by ultrasonication and French press technology. Non-mechanical means other than direct mechanical force to the cell, it can be done by three ways. First one is the using of physical method, second one using of chemical method, and last but not the least enzymatic method. Now physical method means using heat, decompression or giving an osmotic turgor to the cell. And chemical means using antibiotics and detergents, and enzymatic using various lytic enzyme like chitinase we can disrupt the cell. So we have to disrupt the cell first. Now, what are the steps of subcellular fractionation? First step is homogenization. The etymological origin of homogenization, if you look at it, meaning is break open. That means we have to break the cell first. It is concerned mainly with the rupture of the surface membrane. Why homogenization is done? What is the aim? To achieve the highest degree of cell breakage using the minimum disruptive forces without damage to any of the organelle. My objective is to break the cell and take out the organelle intact, and that is done. That is the main objective of homogenization too. Now this homogenizers, they are again of two types. First one is the type 1 homogenizer, 1 for 1. That means the material is subjected to the disruptive force once, and it is using the French press technology. Type 2, the material is repeatedly or continuously exposed to the disruptive force, and it is done by liquid shear. If you recall the previous slide, both this homogenization, whether it is type 1 or type 2, it uses mainly the mechanical method, and mechanical method also comes under which category? Liquid shear, either by French press or other method of liquid shear, that may be ultrasonication. This is the picture of a homogenizer. This picture is of an homogenizer. This is the stand and this is the mortar. Now in this test tube, we have to take the tissue from which we want to extract the cell or disrupt the cell, and it needs to be dissolved in a medium. That medium is known as STKM medium. This have been abbreviated from the first alphabet of the ingredients. S for sucrose with a concentration of 0.25 mole per liter, T for tris, K for potassium, M for magnesium. So these are the important ingredients or components of the solution in which we are going to extract the cell. The optimum pH is 7.4, and it has been the temperature that is utilized for this is 0 to 4 degrees centigrade. We have to maintain this temperature. Why? Because we know that there are various enzymes which are temperature dependent. Enzyme activity is temperature dependent. When we are going to discuss regarding enzymology, we will see how each degree rise of temperature affects its activity. So loss of activity of some of the marker enzymes can occur at room temperature. That's why this temperature is ideal for extraction of the tissue or cell. Next, let us check. Second key step is centrifugation. Now, centrifugation, from the knowledge of your physics, you know it is given as the force that acts away from the center of the rotation, and it is expressed by the formula. Here I have given the formula m omega square r, where m is the mass of the article, omega square is the angular velocity raised to the power 2, and r is the radius from that particular article or substance from the center of the rotation. Now let us check this figure. This blue colored line, this is the axis against which centrifugation will take place, and the centrifugation force that will act away from the axis, already shown by the red arrow, and this yellow arrow, that is for the force of gravity that is acting downward, Next, what we will see? This centrifugation that can be categorized as broadly two types. First one is separation by size, and second one is separation by density. Centrifugation, basically it is a technique of sedimentation. So there will be sedimentation by virtue of its size, that is differential centrifugation. Second type of centrifugation that will go by virtue of its density, that is density gradient centrifugation. Now this density gradient centrifugation I have given in yellow colored question. This is a common catch question in the Viva table. Which type of centrifugation is used for subcellular fractionation? It is density gradient centrifugation. Now this density gradient centrifugation is again of two types. Rate zonal centrifugation, that means where the particle will sediment by virtue of its sedimentation coefficient, and isopycnic centrifugation or buoyant centrifugation, where it will separate by virtue of its property of getting precipitated. These are the two types of centrifugation. Next is which tissue we will use ideally for subcellular fractionation. This figure I have adapted from internet, and this is the monument that is constructed in front of Institute of Cytology and Genetics of the Russian Academy of Sciences, and they have paid a tribute to the laboratory mouse. So here also the ideal tissue or cell for subcellular fractionation is hepatocyte of the lab mouse or liver cell of the lab mouse. Why? Because it offers easy availability, and next is for all the organelles that are present that can be isolated and easily differentiated. That is the main reason. Now what are the steps? So homogenization I have gone. I have disrupted the cell. I have got the mixture of the cellular organelle, and now I have to centrifuge it at higher speed so that by density gradient for each type of speed a particular organelle is separated out. Let us check. So this is the homogenizate, means the after homogenization what is left over is called homogenizate. From here first what I will do, I will centrifuge it at a speed of 500 G for 10 minutes. Fine. So then after that what I will get? I will get, after spinning it, I will get a clear supernatant and a sediment at the bottom. This sediment is nothing but it is the nuclear fraction. Fine. And the supernatant, again I will decant it in another test tube. And this supernatant one, again I will decant it and again I will spin it for 10,000 G for 20 minutes. Here it was 500 G for 10 minutes. Here the speed have changed to 10,000 G for 20 minutes, and again I will get another supernatant and a deposited fragment. The deposited fragment is nothing but mitochondrial fraction. Mitochondrial fraction, and the supernatant 2 is again decanted in another test tube, and that is centrifuged at a speed of 1,05,000 for 1 hour. And here what I will get? Again I will get a sedimented fraction. That sedimented fraction is nothing but the microsomal fraction that will contain the ribosome, the endoplasmic reticulum, the lysosome, and a clear supernatant that is nothing but the cell sap. So this is the 3-step of speeds that are being used ideally for subcellular fractionation. Number 1 speed is 500 G for 10 minutes, 10,000 G for 20 minutes, and 1,05,000 G for 60 minutes. This is the ideal protocol for subcellular fractionation. Now with each of the centrifugation I have got a sedimented fraction, and each of them contain an organelle. Now how much pure they are, that I need to confirm, and that is done by the marker enzyme. Marker means that is particular for that organelle only. So what are marker enzymes? Don't confuse it with biomarkers. This is a common mistake. You get it confused with biomarkers. Biomarkers is something very different. Marker enzymes, put it in your spinal reflex. It is nothing but the enzyme that is required only for identification of subcellular fractionates or organelle. Clear? So it is an enzyme that is localized exclusively in that particular organelle. That is signature for that organelle only. It won't be present simultaneously in many of the organelle. Next, it is useful in monitoring fractionation. I have said I have done a very nice fractionation. How much pure my organelle are, that will be identified by this marker enzyme. Next, marker enzyme also provide information on the purity of the fractionated organelle, and the presence of unwanted marker enzyme activity will give you an idea that that particular organelle is contaminated. Clear? Now, these are the list of marker enzymes. This one is a common question as an MCQ. As per new NMC curriculum, you will have 10 marks of MCQ in each of the paper. So, this is a common question in MCQ as well as memorize this chart. This has also appeared in various NEET PG exam. To start with, mitochondria, succinate dehydrogenase or ATP synthase. Next is the cytosol, lactate dehydrogenase, endoplasmic reticulum, glucose 6-phosphatase, lysosomes, acid phosphatase, cathepsin, beta hexosaminidase, plasma membrane, it is sodium potassium ATPase or 5' nucleotidase, Golgi body, galactosyl transferase or manosidase 2, and for peroxisome it is catalase. So these are the list of marker enzyme. So this is the ideal steps of subcellular fractionation and the marker enzymes in a small summary. Next. we will have an idea regarding what question comes in the exam. Describe the process to separate cell organelles and list the marker enzymes of cell membrane and subcellular organelle. 6 plus 4. Write short note on marker enzyme, 5 marks. These questions have appeared in various university examination of different states. So make a note on this. And next is, these are the references or books which you can go for further study. So please study and make a protocol of subcellular fractionation and note on marker enzymes. This is the first competency.